Greetings. This is lecture number 30. It starts a new series of lectures. This series is on cell signaling. And so in this first lecture, lecture number 30, we'll do an introduction to cell signaling, and then we will end by talking about intracellular receptors. You'll see how that relates. This slide is just a picture of a slide that has been exposed to some sort of signaling molecule, and a dye is used here to quantify the concentration of some second messenger. It may be calcium or something like that. And the red would show where you have a high concentration, the yellow a lower concentration, the green an even lower, and the blue even lower. And so it just shows you that you can have different concentrations of these signaling molecules in a cell, depending on where on the cell the signal binds. So we'll get into all that, but this is really a unit or a section or a series of lectures on how cells do what they do. In other words, or why they do what they do, maybe a better way to put it. In other words, we've talked a lot about different kinds of things that cells do. We will talk about one other things that cells do after we finish this series. And in all those cases, the cell's going to do what it's going to do, oftentimes in response to some sort of signal that comes from outside. So the question is, what are those signals, and how do those signals convey information to the cell and induce the cell to behave in a certain way? In some textbooks, this section would be called cell communication, because that's kind of what we're getting at. In other words, one cell is going to secrete a signal, and that signal is going to be received by a second cell, and that signal is going to tell that second cell what to do. So in other words, it's a way in which those two cells communicate with each other. So you can call it cell signaling, or you can call it cell communication, whatever you want to call it. That's what we're talking about. So let's just kind of look at the way this is going to work overall. You're going to have some sort of signaling molecule. So here's our signal. It's going to be some sort of extracellular signal coming from the outside. On the cell, you're going to have some sort of receptor that receives that signal. Now, in this case, you can see the receptors on the plasma membrane. That may or may not be the case. We'll see. But once the signal engages the receptor, then you're going to have a series of events that take place on the inside, in which some intracellular signaling molecules are going to be generated. So notice you've got an extracellular signal here on the outside, and here you have an intracellular signal. In other words, in most cases, the extracellular signal is not internalized and then serves as an internal signal. Rather, it engages a receptor, and that receptor induces the formation of some intracellular signals. This is called the first messenger, and these intracellular signals are called second messengers. We'll define these in more detail or in more technical terms in a subsequent slide. These intracellular signals then are going to have an effect on some intracellular proteins, and those proteins could do a lot of things. Those proteins may be enzymes, as you can see right here, so we're going to alter metabolism in the cell. Or these proteins may be transcription factors, in which case we're going to alter gene expression. Or they may be components of the cytoskeleton, in which case we're going to alter cell shape or movement. Collectively, you can refer to the proteins that are affected by these intracellular signals as effector proteins. So that's just really a general term, an umbrella term, if you will, that describes all the proteins that are affected by these second messengers. Signal transduction is the process whereby a cell receives this extracellular signal and converts it to an intracellular signal. That's why these are called signal transduction pathways as well, because really what we're going to do is transduce that extracellular signal into an intracellular signal. So that's really what this whole series of lectures. Now let's elaborate on the general scheme that we described in the last slide. Here you can see you've got an extracellular signal binding to its receptor, just like we saw in the last slide. You can also see you've got your effector molecules down here, or your effector proteins over here. And we can see in the middle of this, we've got some proteins that are affected. There are really three things that happen as part of this signal transduction cascade. One is that the signal is going to be amplified. We'll see this in more detail on the next slide, but that's a key part of this. To amplify a signal just obviously means that you want to turn it up. In other words, you could have one extracellular signal that engages a receptor, but you want, if you're a cell, to generate more than one second messenger inside the cell. So that would be to amplify it. The second thing that's going to happen is the cell is going to have to integrate signals. In other words, a cell may be getting two or more extracellular signals binding simultaneously. And in that case, the cell is going to have to take information from one of these pathways and information from the other pathway and integrate that and determine what to do. So information is going to have to be integrated. After all, it's a single cell. It wants to behave in a certain way, so it's going to have to take information from multiple places. And the third thing that's going to happen is you have to distribute this information. So for example, we identified three kinds of effector proteins in the previous slide. We said they could be enzymes, they could be transcription factors, or they could be part of the cytoskeleton. Well, it's possible that a single signaling molecule is going to generate second messengers that affect all three of those effector proteins. So what that means is you would have to distribute that information to multiple proteins. Now, before we leave this slide, you might say, well, what about transduction? This says transduction and amplify. But really, to transduce is just, as we said on the previous slide, to change it from an extracellular to an intracellular message. And then what we're going to do is amplify, integrate, and distribute that message. Now again, let's define second messengers. Second messengers are small intracellular molecules whose concentration changes in response to binding of an extracellular molecule or signal. And it's going to function in one of these signal transduction pathways. In other words, these molecules here are going to be your second messengers. Now let's look at this idea of amplification. We've got the same cartoon on the left that we had on the previous slide, but let's look at the right just to illustrate how important this is. You've got one rhodopsin molecule here, which is going to absorb one photon of light. Now, it doesn't really matter what this pathway is. We just want to use it to illustrate amplification. Well, that one rhodopsin molecule, which absorbs one photon of light, activates 500 G proteins. Now, we'll talk about G proteins later on, but the point is we're activating 500 G proteins, not just one. From that, we're going to make 500 cyclic GMP molecules. 500 cyclic GMP phosphodiesterase molecules are activated. Now, there's no amplification there. 500 G proteins activate 500 of these molecules. But 500 of those molecules result in the production of 10 to the 5th cyclic GMP molecules. Now that's massive amplification. In other words, we've got way more than 500 molecules here. Now those 500 molecules are going to activate 250 cation channels. Specifically, it's going to close them. No amplification there. But massive amplification if you look at the next step, because 10 to the 6th times 10 to the 7th sodium ions are prevented from entering the cell Now, we already know what's going to happen when a sodium ion enters the cell. It's going to change the membrane electric potential because it's positively charged. So the end result here is going to be a change in membrane electric potential. So this is a wonderful example of amplification. It's also a great way to see exactly how these signaling pathways work. Our signaling molecule in this case was light. Okay, our receptor in this case was rhodopsin. And then we've got all of these intermediate steps in here that include the production of some second messengers. And down here, we have a change in cell behavior as a result of a change, in this case, in a cation channel. So this fits perfectly with what we've described so far. So what we will do in this series of lectures is we'll look at some common signal transduction pathways and see how they work in the cell. See exactly what the signal is, see exactly what the receptor is, see what the intracellular second messengers are, and see exactly what happens to the cell as a result of this signaling pathway. Now, we can't look at all of them, but we'll look at some of the classic and common examples that illustrate the way it works. So the last slide had to do with amplification. This one has to do with integration. So again, in this case, what happens is you have two signaling molecules, A and B, binding to two separate receptors. We want to integrate this information. In other words, the target here that's going to be affected is going to be this protein. It's going to get phosphorylated. And that phosphorylated protein is then going to have some downstream effect. But notice that what happens is this is an example of what are called coincidence detectors. In other words, the activation of molecule Y there doesn't really happen unless you have an extracellular signaling molecule that engages its receptor A, and the same thing happens with B. So this is an example of a coincidence detector. In other words, nothing happens unless A and B receptors are activated coincidentally. And it's an example of integration because you're essentially taking information from two different signaling pathways and converging it for a single effect. So that integration is really important, and we'll see some integration. This really kind of illustrates that concept in a different way. But here, let's just go, well, we'll start at the top. This is a cell that's in survival mode. It's not growing, it's not dividing, it's not differentiating, it's not dying. You've got three signaling molecules that are required for survival. So again, you're going to take those three signaling pathways that have been activated, integrate all that information, and the result is going to be survival. Now, it could be that if you have A, B, and C pathways activated and D and E pathways activated, the cell is going to now grow, get larger, and divide. If you have A, B, and C, and F, and G, it may be that that cell doesn't grow and divide, but it doesn't just sit there and survive either. It differentiates. And then in the total absence of signals, you get cell death. So this is another example of integrating information and shows how cells are going to be responding to these extracellular signals simultaneously. This is kind of like what we said with transporters. We looked at transporters individually, but we were aware that multiple transporters had to function simultaneously in order to maintain homeostasis in the cell. The same sort of thing here. The outcome depends on multiple signaling pathways activated simultaneously. Now, this slide is not new at all. This goes all the way back to some of the very earliest lectures. and we were talking about protein phosphorylation and G proteins. The reason we're showing it here is because many of these signaling pathways are going to involve kinases that phosphorylate proteins. We're going to see a lot of protein phosphorylation as a way to activate or inactivate a protein. We're also going to see a lot of G proteins. We already know how G proteins work. We've seen them over and over this semester, so none of this is going to be new. We're just going to look at protein phosphorylation and the use of G proteins and find that they are a key part of these signaling pathways that we're talking about. The other thing this illustrates is that oftentimes there's a balance between activation and inactivation in these pathways. And if you're using phosphorylation as a way to activate a pathway, you can easily see how that balance could be achieved. In other words, we know that protein kinases are going to phosphorylate proteins, maybe turn them on, maybe turn them off. Then phosphatases are going to remove that phosphate. So the balance between the activation of a kinase and a phosphatase is going to be crucial in these signaling pathways. Likewise, for G proteins, we know that if it's got GTP bound to it, it's on. If it has GDP bound to it, it's off. So you're going to have signals then that are going to promote exchange of GDP for GTP. So that's going to, of course, involve GEFs. So we will see many examples of GEFs, and we'll see many examples of GAPs, working in opposite directions to regulate a signaling pathway using classic G protein function. Now, the other thing that we'll see is what's called activation by inhibition. Now, that sounds like a doesn't make any sense. But here's the way that's going to work. If you look over to the left, let's say we have an inactive transcription factor shown in green, and we have some sort of inhibitor protein that binds to it. Well, that transcription factor is going to be inactive. Maybe it's inactive because it's sequestered in the cytosol and can't get to the nucleus, or maybe it's inactive just because it's not active. It needs to undergo some sort of modification to be active. So your upstream signal in this case is going to phosphorylate the inhibitor, and you're going to get gene expression. So that's actually activation by inhibiting, in this case, the ability of the inhibitor protein to do its job. So you can kind of see that what's going on over here. In other words, our signaling molecule is going to activate a protein kinase. That's going to lead to phosphorylation. But that's going to inhibit the protein, the inhibitor protein, which normally inhibits the transcription regulator. So we're going to remove both of these. In other words, we have double negative activation here and here. And that's going to lead to gene expression. So the bottom line here is that the event may not always be straightforward. In other words, it may not necessarily be that a signaling molecule induces some sort of response that leads directly to activation. It could be inhibiting an inhibitor, which is exactly what happens here. By inhibiting an inhibitor, you allow something to happen, and that's why it's called double negative activation. Now, let's switch gears for a little bit and ask a simple question. Where are these signaling molecules coming from? We're not going to worry about the nature of the signaling molecule right now. It's just a simple question, where are they coming from? And I like to start down here in the bottom right because these are ones that we can relate to the easiest, I think. When we think about signaling molecules, this is what you might think of. Here's an endocrine cell that's going to secrete a hormone. So any hormone you might want to think of. Cortisol, estrogen, testosterone, things like that. These are going to be hormones that are secreted by certain cells. These hormones are going to be dumped out into the bloodstream where they can travel throughout the body. As they travel throughout the body, they're going to encounter target cells. A target cell is defined as a cell that has a receptor for that signaling molecule. These hormones are going to encounter a bunch of cells that don't have receptors, and so those cells are just going to ignore the hormone because they can't bind to the hormone. So the definition of an endocrine signaling molecule is that it's a hormone that circulates throughout the body. So the target cell could be anywhere in the body. Now let's go up here straight above that and look at paracrine signaling. In paracrine signaling, your signaling cell is going to release a signaling molecule just like we saw below, except that signaling molecule is not dumped out into the bloodstream where it travels throughout the body. Rather, it functions as a local mediator. It's going to diffuse away from that signaling molecule. So in that case, the target cell has to be pretty close by. The further away the target cell is from the signaling cell, the less likely it will engage the signaling molecule. So that's a paracrine signal. Contact dependent means that the signaling molecule never even leaves the cell that makes it. The signaling cell in this case takes the signaling molecule, puts it in the membrane. The target cell in that case has to be right next to it. In other words, the target cell, as you can see here, has to have a receptor that can engage the signaling molecule, so they've got to be right next to each other. So you can see when you go from A to B to D here, what we're talking about is signaling molecules that are able to affect target cells further and further away. For contact dependent signaling, the target cell has to be right up next to it. For paracrine signaling, the target cell has to be pretty close. And for endocrine signaling, the target cell can be anywhere in the body. Now C illustrates a little bit of a different example and a very specific example. This is what happens with neurons that are releasing neurotransmitters. In this case, it's really kind of like paracrine signaling in the sense that the signaling molecule, or the signaling cell rather, releases the signaling molecule, as you can see right here. The target cell is going to be really close, and in fact they're only separated by a small region, and that region of space is called the synapse. So it's kind of like contact dependent, except you don't have to have direct contact because the neurotransmitter is actually released. It's kind of like paracrine in that it will diffuse, but it's diffusing a very short distance. So those are the 4 different types of cell-to-cell signaling. Now again, in every case, we're going to have what's called signal transduction. We've already used that term, but I've got it on here again just to remind us that that extracellular signal is going to be transduced into an intracellular signal that elicits a certain response. Now before we leave this slide, there's one really important thing to think about. What do all of these have in common? All of these pathways have something in common that you really can't miss. In every single case, the target cell has a receptor. There's a receptor, there's a receptor, there's a receptor, and there's a receptor. So no matter what kind of cell-to-cell signaling you have to have a signal, otherwise we don't have anything to talk about. But your target cell always has to have a receptor. So we'll spend a lot of time talking about the signaling molecules and the receptors. So this slide shows another important aspect of these signaling pathways that we have to consider. So if you look over on the left, you'll see a particular signaling molecule. In this case, it's acetylcholine. And if you look to the right, you'll see a heart pacemaker cell. So these are cells in the heart that determine the rate at which the heart contracts. So here's acetylcholine. That's your first messenger, your signal binding to its receptor protein, the acetylcholine receptor. And when it binds, look what happens. If you look at this EKG readout down here and compare this to this, you can see that contraction has slowed down. So we have decreased firing. Now, if you look to the right of that in the salivary gland, you'll notice that you also have acetylcholine binding to a very similar, maybe not exactly, but a very similar acetylcholine receptor. But look at the outcome. The outcome here is totally different. You don't get a change in the firing rate because that's not what these cells do. These are salivary gland cells. So these cells have secretory vesicles. We know all about those. And these secretory vesicles are going to fuse with the membrane, so we've got exocytosis going on here. And that's going to dump these the contents of these secretory vessels out into the extracellular space. In this case, they're going to end up in your mouth, okay, as part of saliva. Now, if you look over to the right even further, you see skeletal muscle, and you see the same signaling molecule binding to the receptor on skeletal muscle. And in this case, what happens is we're going to increase contraction. So the skeletal muscle is going to contract. So if you think about what's going on here, it's kind of interesting. In the first two examples with the heart pacemaker cell and the salivary gland cell, you have the same signaling molecule binding to very similar receptors with two totally different outcomes. How can that be? Well, the reason for that is because the sequence of events, the signaling events, and or the target proteins that are affected are totally different. So just because you have the same signal binding to the same receptor doesn't mean you're going to get the same effect in every cell. It depends on what the target proteins are in that particular cell. So you can have two drastically different responses as we've got here. One with changing the rate of contraction and one with inducing secretion. Now, if you look at skeletal muscle, there's another thing to consider. Here, you've got the same signaling molecule. You have a different outcome, but that's not really the point here. The point here is that you're binding to a different receptor. It turns out in this case, these receptors are called G protein coupled receptors. Now, we'll talk about those in great detail. This receptor is not a G protein coupled receptor. This receptor is actually an ion channel. So there's something really important to consider here, and that is when we say that the signaling molecule binds to a receptor, that receptor can have another function. In this case, the receptor is an ion channel. So now you've got a different receptor and a different response. So what's the take-home message from this? The take-home message is that receptors don't always have to be the same for a given signaling molecule, and the outcome in the cell doesn't have to be the same. So if you want to know what receptor is engaged and what the outcome is when a cell is exposed to a particular signaling molecule, you have to look at that cell. You can't draw a generalization. Said another way, these pathways are very specific. Signaling molecule function is cell type dependent. Another general concept that we need to talk about is this concept called crosstalk. What is crosstalk? Well, crosstalk is it's kind of like integration, but not exactly. In this case, we've got two different signaling molecules. One is engaging a G protein coupled receptor. One is engaging a receptor tyrosine kinase. We'll talk about those later, but the point is they're receptors. And notice that these down here in this region are the second messengers that are generated by these pathways. Now, crosstalk refers to the fact that these pathways talk to each other. That's kind of what it means. Okay. So, for example, this phospholipase C right here can be activated by a receptor tyrosine kinase. It can also be activated by a G protein here. And you can see, most importantly, when it comes to crosstalk, is that let's say that this pathway activates, the receptor tyrosine kinase pathway activates that MAP kinase. Well, that MAP kinase is going to affect target proteins over here, but it's also going to affect transcription regulators over here. This calcium-dependent kinase right here is going to affect target proteins over here and transcription regulators here. So it's kind of like these pathways have to talk to each other in order to have the desired response in the cell. So the transcription regulators that are activated over here to the left can be activated by signaling molecules that bind to the G protein coupled receptor, and also by signaling molecules that bind to the receptor tyrosine kinase. Another general concept that we have to consider is how quickly or slowly these signaling pathways take to have their effect, and also whether you have long or short-term effects. Now, let's just think about a signaling molecule here that engages a receptor. That's what we've got going on here. Let's just say that this is going to activate some sort of transcription factor. So if it activates a transcription factor, we know that we're going to upregulate expression of this gene. So we're upregulating expression of that gene. We're transcribing that gene, processing the RNA, and we're going to make protein, let's say more of a particular protein. That protein now, could be an enzyme or something like that, is going to have some effect inside the cell. But ask yourself how long that's going to take. You don't have to put a number on it, but it's not hard to imagine that's going to take a little bit of time, right? In other words, not only do you have to activate that pathway, but you have to decondense the gene, you have to transcribe the gene, you have to process the mRNA, you have to export the mRNA, you have to translate the mRNA, you may have to process that protein, all before you get the desired effect within the cell. So any signaling pathway that alters protein synthesis is going to be a relatively slow pathway. It's going to take minutes to hours. So what's the opposite of that? Well, that would be a pathway that acts quickly, and that's going to happen when you've already got a protein that's present in the cell. So none of that we described has to take place. You don't have to transcribe the gene or process the mRNA or anything like that. You just activate or inactivate the protein, as the case may be. Those are really fast pathways. Because you don't have to make the protein. You just change the activity of a protein that's already there. These slow pathways, these pathways in which protein synthesis is altered, tend to be long-term pathways. Now this kind of makes sense because if you transcribe a gene, process the mRNA, export the mRNA, you're going to be making new protein from that mRNA as long as that mRNA is around. So as we know from previous lectures, that's going to relate to the stability of that mRNA. The point here is that it's called a long-term pathway because you could be having effects in the cell taking place long after the signaling molecule is gone. In other words, the effects are really going to be determined by the concentration of the protein, and the concentration of the protein is going to be determined, at least in part, by how stable the mRNA is. In contrast, these fast pathways tend to be short-term pathways. In other words, you alter a protein's function, it's going to affect some sort of thing in the cell. Let's just say you alter that protein by phosphorylation. Well, you could rapidly dephosphorylate that protein as well. So these tend to be really short-term. So you have fast responses that tend to be short-term, and you have long responses that tend to be slow. Now, let's think about the turnover rate of these intracellular molecules. This is really an important thing that determines the outcome in a cell. So this is a little bit of a complicated slide, but you've got some sort of intracellular molecule, some sort of second messenger that you've generated. So the number in blue is the half-life of the protein. We're going to say that this intracellular molecule is a protein. Now, we know what half-life is. Half-life is the length of time it takes for half of the protein to disappear. So a long half-life would mean a stable protein that hangs around for a long period of time. So over here on the left, we've got 5 different, let's say, proteins with different half-lives, ranging from 200 minutes down to 1 minute. On the x-axis, you have minutes after the synthesis rate of the protein has been decreased by a factor of 10. And over here, you have the relative concentration of that molecule. So let's think about what happens if you've got a molecule. We'll just arbitrarily say its concentration is up here at 10. Let's use yellow here. Okay. It's got a long half-life, a 200-minute half-life. That means that's a very stable protein. It's not being degraded at a rapid rate. Now what the bottom shows is that you've decreased the synthesis of that protein by a factor of 10. Well, what's going to happen to the concentration of that protein if you decrease the synthesis of it? Well, if it's a protein with a long half-life, not much is going to happen. On the other hand, if you've got a protein that has a really short half-life, if you stop making that protein, the concentration of that protein is going to drop dramatically. So that's what's being shown here for this one. Okay? In other words, in this case, what's happening is because that protein has a short half-life, it turns over rapidly. If you stop making it, the concentration disappears or goes way down. Now there's a flip side to this. If you look on the right-hand side, this is minutes after the synthesis rate has been increased by a factor of 10. So let's do the same thing. You've got a protein that has a half-life of 200 minutes. You increase the synthesis rate of that protein. How much is the concentration changing? Well, it's not going to change a whole lot. It's not going to change a whole lot because the concentration is essentially pretty much steady. Okay, so in other words, if you've got a high concentration of a molecule and you add a little bit more to that, you don't really change the overall concentration. Now it's going to go up a little bit, but not much. But on the other hand, consider the opposite, or the other end of the spectrum, we should say. You got a protein with a short half-life. It means it's going to turn over rapidly. Now, if you increase the rate of synthesis, you're really going to increase dramatically the concentration of that protein. So what's the bottom line here? The bottom line is proteins with a short half-life, the concentration can change dramatically. But if you got a protein with a long half-life, the concentration is pretty stable. So what matters in these signaling pathways then, in terms of the effect in the cell, is not only whether or not you have the signaling molecule, but what also matters is whether or not, or how stable the intracellular messengers are. Now there's another thing you got to consider here. If you have an intracellular messenger with a long half-life, you can have effects in the cell long after the signaling molecule is gone. But if you've got an important second messenger with a short half-life, then you're going to have to keep the signaling molecule around to continue activating that pathway in order to have the desired effect. Now, a couple of other general things we need to consider before we look at some specific examples. Cells are going to adapt to these signals. They're going to be desensitized. In other words, how's the cell going to respond to the presence of these signals? Well, let's look over here on the far left. Look at what happens here. You have a receptor or a signaling molecule that engages a receptor. Now we've already seen how this can happen. That's going to, in this case, it's going to be internalized by an endosome. And that receptor can actually sit there in that endosome. Eventually it's going to go back to the cell surface through a recycling endosome. But it can be temporarily sequestered in that endosome. So this is a way to desensitize the cell to an extracellular signal. In other words, if you don't put the receptor back on the surface of the cell, it's not available to respond to more signals. Now, a more dramatic example of that is you could actually take that receptor and degrade it in lysosomes. Now we know in that case, this is probably going to be tagged with what? We would have ubiquitin over here, ubiquitin, and it would be tagged, degraded, starting in the late endosome in that multivesicular body. Another way you can do this is instead of moving the receptor off the plasma membrane, you can inactivate it. So it could be that when the signaling molecule binds to the receptor, you get simultaneous inactivation. Now that may be counterintuitive because you're trying to activate a pathway, but this is the analogy that we've used over and over this semester. It's the light switch analogy. In other words, if you really want to carefully control the cellular response to this, you activate the pathway, you simultaneously inactivate it, and it's the balance between those two that determines the end result. In this example over here, you've got your intracellular molecule. Now this one is similar. Let's number these just for the sake of discussion. You have 1, 2, 3, 4, and 5. So 4 is like 5, except that you're not inactivating the receptor itself. In this case, you're inactivating an intracellular signaling molecule that's been generated. The end result is going to be the same. Okay. And what about in case number 5? In case number 5, what's going to happen is you're going to, as part of the pathway, you actually activate some sort of inhibitory protein. And by activating that inhibitory protein, you're going to shut the pathway down. So in all these cases, what's happening? You're activating the pathway, but you're simultaneously engaging some sort of mechanism or activating some sort of mechanism to shut the pathway down. This ensures that these signaling cascades or transduction pathways don't go haywire. So you've got to check on the system, and it can happen in one of these different ways. So this allows cells to detect percentage changes in a signal over a wide range of stimulus strengths. Okay, in other words, no matter what your stimulus strength is, we can detect small percentage changes. Again, this is the light switch analogy. So we could have a high or a low concentration of signaling molecule, but we can get small changes, or we can control the changes, not only by controlling the amount of the stimulus molecule or the signaling molecule, but also by controlling the receptor. So we can fine tune this even more. That's what that sentence is intended to mean. We can fine tune the system even more. Okay, now we're going to start the process of looking at some specific pathways. If we go back to the beginning, remember that we're always going to have a signaling molecule, and we're always going to have a receptor. Now, in most cases, the receptor is going to be on the surface of the cell, and that's exactly what you got going on here. It's an extracellular signal after all. But there are some signals that actually can cross the membrane. Now, we just finished lectures on things crossing the membrane. So if something's going to cross a membrane by diffusion, by not binding to a receptor, what are the characteristics? Number one, it's going to have to be small. And number two, it's going to have to be relatively nonpolar. So what if you have a signaling molecule that's a small hydrophobic signaling molecule? Well, it can cross the membrane. If it can cross the membrane, does that mean you don't need a receptor? No. It just simply means that the receptor can be an intracellular receptor instead of a plasma membrane receptor. Now, the other thing is, because these are hydrophobic signaling molecules, they're often going to exist in the extracellular space bound to some carrier protein, because you can't just have these hydrophobic signaling molecules out in the extracellular space, or they would aggregate, because we know that's what nonpolar molecules do in an aqueous environment. So often they're going to be associated with some sort of carrier protein. Once they reach the membrane, the molecule diffuses across the membrane and then binds with an intracellular receptor. Over here, you don't need a carrier protein because this is a hydrophilic signaling molecule, so it's perfectly happy in the aqueous environment. Now, most of what we're going to talk about in the subsequent lectures is going to involve cell surface receptors. So what we're going to do is talk about intracellular receptors for the rest of this lecture, kind of get those out of the way, because they're kind of a specialized case. Here are some examples of hormones that function this way. All of these hormones have intracellular receptors. And what do you notice about these hormones? All of them. They're small and they're relatively nonpolar. Now, I recognize they've got some polar groups on there, but by and large, these are nonpolar molecules. The other thing that you'll notice about a lot of these molecules is that they have a structure that's very similar to that of cholesterol. So it brings up an interesting question. Is cholesterol a signaling molecule? Turns out cholesterol is not a very effective signaling molecule at all, for one simple reason. It's everywhere. It's already in membranes. It's an important component of membranes. So that kind of precludes its use as a signaling molecule. Signaling molecules have to be present under certain conditions at certain times. So you can't have a ubiquitous molecule like cholesterol serving as a signaling molecule. But anyway, cortisol, estradiol, testosterone, vitamin D3, retinoic acid, and thyroxine are all examples of small nonpolar molecules that can cross the membrane and serve as signaling molecules. Now, how are they going to work? Well, let's use cortisol as an example. Here's cortisol crossing the membrane. Now, in the case of cortisol, it turns out that here's its intracellular receptor. This is called a nuclear receptor protein, because what happens is when cortisol crosses the membrane and engages the receptor, it induces a conformational change, and that receptor now, in complex with the cortisol, is translocated to the nucleus, where it binds to a cis-regulatory sequence. So it's going to bind right there to some sort of cis-regulatory sequence. Okay, and we're going to activate transcription. Now, by the way, let me just ask by way of review here, or just to think about, what do you think this conformational change is going to do here? In other words, if that nuclear receptor protein is in the cytosol, but once it binds to cortisol, it ends up in the nucleus, then what has probably happened? You've probably exposed a nuclear localization signal. Maybe the nuclear localization signal was buried on the inside of the protein and now it's exposed on the outside of the protein. Or maybe the nuclear localization signal was covered up or masked by some other protein or some other domain of this protein and it gets moved out of the way. But in all likelihood, you're going to expose a nuclear localization signal, and that's going to allow it now to be translocated to the nucleus. So there's a whole family of transcription factors called the nuclear receptor superfamily of transcription regulators. They're all going to work basically the same way. Okay. Let's look at another example of these nuclear receptor superfamilies. This is an androgen receptor. An androgen is any hormone that gives an organism male sex characteristics. Now, this is really cool to think about how this works. I'm going to take the highlighter here and I'm going to cover this portion up. Okay, ignore that portion for just a second. Now look at what you've got there. You have an activation domain and a DNA binding domain. What do you have? That's a classic looking transcription factor. Those are the components that we said transcription factors have. Well, it turns out these receptors, in many cases, are transcription factors, because that's clearly what that is. But they're transcription factors with another domain. The third domain is the ligand binding domain. That's the domain that binds the second messenger. Now, in this case, what happens is the androgen is going to come in. It's going to bind to the binding site. That induces a conformational change that includes the removal of this inhibitory protein. So the inhibitory protein is released. That conformational change then also changes this protein in such a way that this region right now becomes a binding site for a coactivator. So in this case, unlike cortisol, it's not so much that the transcription factor, in the case of cortisol, needed to be translocated to the nucleus. In this case, your transcription factor is already in the nucleus. It's just not active. And it's not active because it has this inhibitory protein bound to it, and it can't bind coactivators. So if you look at the bottom, you can actually see this kind of conformational change. So over here, you have no ligand bound. Here, you have the ligand bound to the binding site. And that now is going to allow the alpha helix of a coactivator to bind. So this androgen receptor is called a ligand-dependent transcription factor. So this is really cool. This takes us all the way back to the regulation of gene expression. We know how that works. We know how important transcription factors are. Now what you see is some transcription factors actually double as receptors for signaling molecules. I also like to compare the cortisol to the androgen situation, because what you can see in both those cases is that we can affect transcription factors by either altering their location, cytosol versus nucleus, or in this case, by altering their structure and therefore their function. Now we're going to look at another example of a signaling molecule that can cross a membrane. sometimes signaling molecules that cross membranes are not small hydrophobic molecules like we saw in the last group, but they're gases. And the best example of this is nitric oxide. So let's take a look at what's going to happen here. Now, before we do this, we've kind of got to look at this blood vessel over here. So these are endothelial cells. I'll outline an endothelial cell lining a blood vessel. So this is the lumen of the blood vessel. This is where blood would flow. Okay. Now, underneath those endothelial cells are smooth muscle cells right here. Those smooth muscle cells can contract, and if those smooth muscle cells contract, then the lumen of that blood vessel gets smaller, and you're going to constrict the flow of blood. If those smooth muscle cells relax and the lumen gets larger, you have enhanced blood flow. So let's take a look at what happens here. Here's acetylcholine binding to the acetylcholine receptor right here. We're going to induce a sequence of events that's going to lead to the production of inositol triphosphate. We'll get to this pathway later on. The release of calcium from the sarcoplasmic reticulum. The activation of an enzyme called nitric oxide synthase, which converts arginine into nitric oxide. The nitric oxide diffuses across the membrane, as we know that it can do because it's such a small molecule. It can also then diffuse across the membrane of the smooth muscle cell. Once it gets into the smooth muscle cell, it's going to bind to guanylyl cyclase. Now, guanylyl cyclase is going to convert GTP to cyclic GMP. Okay. So the cyclic GMP then is going to lead to smooth muscle cell relaxation. Okay, and that smooth muscle cell relaxation is going to increase the diameter of that blood vessel and increase blood flow. Now let's look down here at the bottom and we'll just see this in a little bit more detail. Here's GTP. So this is what we're looking at. There's nitric oxide binding to guanylyl cyclase. We're converting it to cyclic GMP, and it's that cyclic GMP that leads to smooth muscle cell relaxation. So in that case, what is your signaling molecule? Your signaling molecule is a gas. Okay. So this is a little bit of a different situation because it turns out that the receptor, in quotes, for the gas is actually the enzyme that performs the function of converting GTP to cyclic GMP. Now there are two well-known examples of how this works, and I think they illustrate this really nicely. Think of somebody that has angina, so chest pain due to constricted blood flow to the heart. Oftentimes, to relieve that angina, they'll take nitroglycerin. So nitroglycerin will be converted to nitric oxide, and the nitric oxide then will do exactly what we've described in this pathway. We're going to increase smooth muscle cell relaxation. That's going to cause an increase in the diameter of blood vessels. We're going to get increased blood flow, and the chest pain is alleviated. Now there's another well-known example of this. There's actually a drug that everybody's familiar with that is affected by this pathway. It turns out that cyclic GMP is degraded by an enzyme called phosphodiesterase type 5. So let's think about that for just a second. If you inhibit phosphodiesterase type 5, you're going to have continued elevated concentrations of cyclic GMP. You're going to have continued smooth muscle cell relaxation and continued blood flow. Well, it turns out that ED drugs, erectile dysfunction drugs, such as Viagra, work in this way. It turns out that Viagra is an inhibitor of phosphodiesterase type 5. So by inhibiting phosphodiesterase type 5, you maintain relatively high concentrations of cyclic GMP. the high concentrations of cyclic GMP lead to an increased diameter of vessels in the penis. That's going to increase blood flow, and that's going to allow an erection to be maintained. That's the basis for those erectile dysfunction drugs. So they actually are interfering in some ways with the breakdown of one of these final products that results from a gas, in this case nitric oxide, serving as part of a signaling pathway. Okay, so that summarizes this lecture, and you can see what we've done is we've talked about intracellular receptors now and some general considerations. For the rest of this series of lectures, we will focus on cell surface receptors.